Hello to all the HubLE audience. My name is Fatemeh Malekipour. I'm a postdoctoral research fellow at the University of Melbourne, Australia. I'm also a member of the Meet the First Author Committee at HubLE. I'm here today for an edition of Meet the First Author with Taraneh Rezaee. We will be talking about their paper titled Increasing Fluoride Content Deteriorates Rat Bone Mechanical Properties, which was published in the Bone Journal. So welcome, Taraneh. Welcome to HubLE. And to start with, could you tell us a summary of your work and how it fits into the big picture of this field?
Thanks to you and HubLE for giving me this opportunity to take part in this interview. The general idea of this paper was we already knew that elevation of bone fluoride levels due to drinking beverage with high fluoride content can result in a skeletal fluorosis. Skeletal fluorosis is a metabolic bone disease, and it can lead to joint pain and skeletal deformities and also fracture. We know that fluoride can alter bone mineral composition, and so it's likely to affect bone tissue properties and consequently whole bone mechanical properties. To determine this hypothesis, we incubated some rat bone samples in different doses of sodium fluoride. And then we assessed bone mechanical properties using a CRPI method and also traditional three-point bending method. We also determined tissue geometry and also tissue mineral density by a micro-CT device. And finally, we found that in vitro sodium fluoride incubation can deteriorate bone mechanical behavior in both tissue level and whole bone level.
Well, that's great. Would you like to share any figures and methodology with us, if you have got any?
So in this picture, you can see that we incubated some ulnae from the rat, female rat actually, in different doses of sodium fluoride, and then we assessed the mechanical properties of those incubated bone, and we finally measured the fluoride concentration for the treated bone samples.
Okay. So what was the main results? What did you found from this measurements?
Actually, since we observed that bone tissue which has been treated with fluoride has really poor mechanical properties, we learned that a high dosage of fluoride can really affect the people around the world who are drinking high-level fluoride beverages.
So in these mice, how high is the fluoride that can actually affect the properties of bone? Did you find a threshold for this value, or what did you find basically?
Actually, it was kind of in vitro incubation. That's a little different with the in vivo study. Right. We incubated the samples in 0.05 molar to 1.5 molar. The threshold for a skeletal fluorosis based on the WHO report, it's 1.5 ppm fluoride in the drinking water usually, right. And some papers confirm it as 4 ppm for fluoride, but our dosage was were kind of in vitro incubation.
And I suppose for a rat, it's probably different with human, is that right?
Of course, it would be different with human.
Right. Okay.
Yes.
So with these limitations, what do you think that is the basically the findings of the paper? What does the findings tell you?
Actually, we try to mimic the skeletal fluorosis using in vitro incubation in fluoride, and there are two aspects. First, we know that skeletal fluorosis is an endemic bone disease, and it can affect millions of people worldwide who are consuming water or tea or average, including high fluoride concentration, specifically population in South and East Asia, Africa, Middle East, or even some places in Australia and United States. However, there is a limited data about the biomechanics of the fluorosis bone due to the limited available bone samples from those patients, because we don't have access to many samples from the human who are suffering skeletal
fluorosis. Yeah.
So,
so basically, you're looking at the mechanism rather than threshold.
Yeah, we hope the result of this study would be the initial step for simulating the skeletal fluorosis using in vitro incubation in sodium fluoride instead of using real bone. And we hope, we can provide this opportunity for researchers in bone area everywhere to investigate the effects of this endemic fluorosis on bone biomechanics. And we hope it can help designing future in vivo animal studies as well.
Oh, okay, yeah, that would be awesome. Great. So the other question I had was, what was your experience working with rat? And you studied rats in this, right?
Okay. Yeah. So actually, they were some small samples, and when we incubated the samples in fluoride, then to confirm how much fluoride it actually uptake, the bone, how much fluoride actually uptake. So we had to measure the fluoride concentration using the ion probe, fluoride ion selective probe, and it was a new protocol, brand new protocol for me. That was my first time that I tried to use this protocol, and as I mentioned, the samples were very small. I did a lot of literature research to understand how this protocol worked, and it was kind of teamwork. My lab mates, my mentor helped me a lot to accomplish this goal, and we finally measured the fluoride content for all the samples.
Okay, yeah, great job.
Thank you.
Awesome. So what is next? What do you think is for the future? Do you aim to do more similar studies or in vivo, or what's in the future?
Actually, CRPI, or cyclic reference point indentation, which we had used for our samples, is a relatively new method for investigating the bone biomechanics. The option of this device enables physicians to in vivo determine the bone properties in live patients as well. So we hope that this study can help the physician in the future, and this study supported this idea that CRPI may serve as a useful tool to measure material properties of bone, and we hope the result could be useful for designing such future studies about the bone fragility due to a skeletal fluorosis, and maybe it can help physicians to better understand bone properties of live patients in earlier stage of a skeletal fluorosis.
Yeah, awesome. Great. Thank you very much, Taraneh, for the time and discussing your work with us today. That was great. I hope we see a lot more from your group and yourself about these studies. Thanks to all HubLE audience for taking the time to watch this video. And if you would like to get involved with Meet the First Author, please contact us. Thank you very much.